Thanks for the organisers for giving me the opportunity to talk about some work we've been doing in our labs in Cardiff and with collaborators. So I'm going to talk about stem cells in healthcare, and I'm going to focus on two areas, because basically if you look at the potential role of stem cells in healthcare, they fall into two areas. You can either use cells as tools, and a lot of people are interested in that. Specifically, does stem cells give you the opportunity to make better drug discovery models, either for looking at drug efficacy or, as I'm going to show you, for drug safety, better prediction of drug liabilities, and then obviously cells as treatment. So I'm going to talk about two examples. Coincidentally, they both come out of collaborations with groups in San Francisco. One, I'm going to talk about looking at cardiotoxicity of anti-cancer drugs, and that's a collaboration we've been running for some time now with Genentech in San Francisco. And then the second example relates to stem cells in therapy, an important area in looking at expansion of cells. Both of these are linked together by the use of high-throughput, high-content imaging, cellular imaging. So basically the concept of using automated microscopy, if you like, to generate very large amounts of data, which we believe is a very efficient way of looking at cell biology, because you're doing it in situ and therefore you're doing it in context. And that allows you to find things that you can't find by doing conventional investigations. So let's look at the use of stem cell-derived cardiomyocytes in this example in cardiotoxicity. So why did we choose to do this? And why looking at anti-cancer drugs? Well, as you can see there, oncology drugs, it's the largest pipeline in the pharmaceutical and biotech industry today. And cardiotoxicity is a well-known issue in drug development in general. It's the leading cause of drug withdrawal and failure, and it's particularly prevalent in anti-cancer drugs and in cancer patients. And these are a number of, in this case, tyrosine kinase inhibitors with reported cardiotoxicity. And these are amongst the panel of drugs that I'm going to show you. And cardiotoxicity can arise for a number of reasons. You get, as shown up here on this side, classical small molecule off-target effects where a kinase inhibitor can be more or less promiscuous. So it may hit its target, but also go off and hit another kinase. Increasingly important, I believe, in the future as we go to the development of more specific drugs, particularly those like Herceptin based on recombinant antibodies, you're not going to get off-target effects from something which is exquisitely designed only to go for its human target. Great, no off-target effects. Problem is you get off-therapy effects because, kinases or other proteins within cells generally don't do just one job. They'll do the same job in different tissues or different jobs in different tissues. So the toxicity of Herceptin is a classical example. It's specific to a protein, does one job in this cell, but you don't want it to interfere in this cell. So in terms of, what's the role of stem cell-derived cardiomyocytes, hepatocytes, whatever, in drug discovery, this is how I see them. They're very much as an integrating role, and they're a game changer. So if we look at cardiotoxicity, there are many, many ways in which a drug, a compound, can impact on a cardiomyocyte in the heart to affect its ability to be able to beat. Most people think of electrophysiology as the first thing that comes to mind, HERG, right? So drug binds to a potassium channel, affects the ability of affects the length, the action potential, affects the ability of that heart cell to beat. But in fact, and AstraZeneca did a study of 1,000 failed compounds. Only 25% of those compounds interact with the electrophysiology of the cell. 75% interfere with mitochondria, calcium handling, or something else. So basically, you need to have this central platform surrounded by the best analytical platforms to get a holistic surveillance of all the potential liabilities of a drug in cardiotoxicity. And that's where I think this stem cells, and now us and others are producing industrial quantities of these stem cell-derived cardiomyocytes. They're very much a game changer. So I'm going to talk about the use of high-content analysis to look at functional and structural toxicity. This is the panel of drugs. There's around 140 here. Most of them are kinase inhibitors. They include drugs in development, failed drugs, and drugs which are still on the market. And what we're looking at by imaging are very important things like membrane integrity, mitochondrial function, calcium homeostasis. Impact any of those, and cardiomyocytes lose their ability to function properly. So this is the schematic of what we do. We take cardiomyocytes, we incubate them with a large dose range of compounds over different time periods, so we're getting both acute and chronic exposure. We image those, we perform image analysis, and we get shed loads of data. You're getting gigabytes or terabytes of data, which gives us a similar problem to the genomics guys of how do we store that, when do we decide to throw it away, et cetera, et cetera. We basically don't throw it away, we just keep buying new servers, and our IT people shout at us. So this is, for a biologist, I'm a cell biologist. Cell biologists like to look at pictures before they look at the numbers. This is what you see. So this is valtalinib, low dose, red is mitochondria. They still look fairly healthy here. Green is intracellular calcium, low levels. They haven't been affected yet. As you go up the dose-response curve, you see a drop in calcium homeostasis and a decrease in mitochondrial integrity. All right? That's how a cell biologist describes it. But to look at large numbers of cells, you need numbers. You need quantitative data, and this is what high-content analysis gives you. So this is the data from all of the compounds. We take multiple parameters, some of which relate to mitochondria, some to calcium, some to other parameters. We construct these profiles, and you can see they're colored by dose. And you can see some of them don't change, some of them change quite dramatically. So I can look at that and say, well, that's okay, that one isn't okay. But an unbiased way is to use the same way you basically look at microarray data, which is to cluster it. This is SOM clustering, but you can do it by k-means or whatever. So this is at low dose. Here's the negative and positive controls. Nothing's happening here. And if you go up the dose-response curve, you see there's a positive control. So these are the toxic drugs. And if we do that, we can basically take that panel of drugs and put them into bins and classify them into whether they're toxic, moderately toxic, or non-toxic. And not just whether they're toxic or not, but also the mechanism by which they're toxic, which is obviously important. If you do that early in the drug discovery process, you might be able to do something about it. Keep the efficacy, drop the toxicity. So that's all fine and dandy, except the heart isn't as simple as that, right? So how many people had a cooked breakfast? How many people will admit to having a cooked breakfast? All right? How many had a cake with their coffee? Okay? So those who had a cake with their coffee, your heart is metabolizing slightly different from those who didn't have a cake with their coffee, okay? Important thing is, heart cells are chock full of mitochondria. Why? Because they need tons and tons of ATP. They make ATP in different ways. They can make it through glycolysis. They can make it through heavy use of the mitochondria through oxidative phosphorylation. A lot of drugs impact on mitochondria, and the degree of that impact is going to be determined by how hard those mitochondria are working at any given time. So what we did was repeat that screen, and instead of using glucose media, which the previous screen used, we used glucose media and galactose media. In galactose media, cardiomyocytes can't go through glycolysis because they get a net gain of 0 ATP. They have to use oxidative phosphorylation and glutamine as a substrate. Their mitochondria are working hard. If we do that, some of the compounds show different effects, and I've shown that by deltas up here. I haven't got time to go into this, but you can see here, this is glucose, this compound mibrutinib, does nothing to cells operating glycolytically. Cells in oxidative phosphorylation is lethal. So you can see this here. Here's the mitochondria, totally unaffected, very dramatically affected. And you can see that here. This is an oxidative phosphorylation. That's a toxic profile. That's a non-toxic profile. And what we found is by using differential analysis of different parameters, we can see that that drug is in fact impacting directly on mitochondrial integrity, way, way before impacts the viability of the cells. So by using that sophisticated analysis, you can pick out that compound as being a mitochondrial toxin, and we confirmed that by respirometry and other measurements. So what is magrotinib? It's an irreversible ERB2 RTK inhibitor developed by Takeda some time ago. They only ever published on its efficacy. They did a phase 1 clinical trial, never reported the results. You can draw your own conclusions. But I would advocate that had they had that data, which I just showed you, they wouldn't have got to that stage and would have probably saved an awful lot of money. So that's one use of stem cells in toxicity. Second one, I'm going to talk about using the same screening techniques looking at stem cells for therapy. So why are we interested in this? So one of the key issues in any cell therapy is how do you get enough cells to form an effective dose, right? It's a lot of cells. So in this case, we have several collaborations with groups in the US and in Europe looking at expanding T cells for immunotherapy in cancer. Essentially, for autologous therapy, taking cells out of melanoma tissue, expanding up those tumor-infiltrating cells, and putting them back into the patient to allow their immune response to attack that tumor. So you're talking about 20 billion cells in a dose or more. And this is a wave bioreactor. It's a method, a contained method of growing very large number of cells, which is very important if you're going into GMP, where you need to have parallel processing of different patient samples. So this example is about expansion of HSC cells. And it's a collaboration with Andy Levitt and Michelle Arkin's lab in the US and our lab in Cardiff. And what it's about is expanding HSCs from cord blood, right? So freezing cord blood, a lot of people are doing it now. Cord blood banks are being established, expanding their stocks. So cord blood has the advantage, it's non-invasive, it's high availability, so you don't have to drill into somebody to get bone marrow out, to get a bone marrow transplant. And there's improved mismatch tolerance. Problem is, you don't get much cord blood. There's not many stem cells in cord blood. So what people are interested in is methods that they can start with cord blood, expand that up to keep growing those cells, either for autologous therapy or eventually for allogeneic therapy, where you need huge amounts of pooled cells. And what we did was basically to set up a screen to look for molecules which would modulate the proliferation of stem cells. Now, screening was just alluded to by the previous speaker. Screening means many things to different people and different things to different people. 10 or whatever years ago, the standard paradigm for screening was you do as many compounds as possible with the simplest assay as possible. Basically, you dumb everything down to bring the cost down and bring the numbers up. So you get a cheap screen, but what's the value of it? Nowadays, people, because these technologies, the type of imaging technologies that I've just described are available, people can do screens which are more meaningful. And in this case, you're doing a screen on primary cells, which is not trivial to do at a large scale. But it works. So this is the project plan. So basically doing assay development, high throughput, high content screening, validate the compounds coming out of those screens by xenograft transplants. So basically that's the gold standard to prove that the cells you've expanded still have the capacity to reconstitute the immune system. Then looking at the mechanism of action, and then taking those compounds into our bioreactors to look at methods to use those compounds, either alone or in combination, to expand HSCs. So this is where we are. The guys in California have done over 126,000 compounds, 510 hits, and they're going through confirmation, dose response curves, repurchases at the moment. So a very complicated assay to do to get cells, primary cells, which don't stick very well to plastic, to work in a 384 well assay and robustly generate hits. So this is the data from the screen. And you can see that out of that, we've got a number of hits which are beginning to show similar chemical structures. So we believe those are genuine hits. So I think hopefully I've shown you two concrete examples of using stem cells as tools for drug discovery and beginning on the process to develop methods to expand cells for cell therapy. So I'd like to thank our collaborators at Genentech, USF and QB3, and the guys back in Cardiff, and thank you for your attention.